In humans, it's important because preformed clinically significant antibodies in the ABO system exist. And essentially, the antibodies are made after about 4 months of age through exposure to gut flora. So they're there, they're clinically significant, and when you actually do the blood group, and you can see on the tile or in the tube the instant agglutination of those red cells. And for me, that's always been a wow. It's always been a wow. Screening for antibodies to other red cell antigens is also performed because if transfused, red cells are destroyed by the patient's antibodies. And then the life of those transfused cells is shortened, and the patient is compromised, sometimes in a life-threatening manner, by the destruction of the red cells and the release of the breakdown products. So the ABO blood group is important because it is the antibodies are clinically significant. Other antibodies are formed through exposure by pregnancy or transfusion or intravenous drug use with shared needles, and they can also destroy red cells, and that, if you're doing a transfusion, that's not what you want. You want the transfused cells to last for the expected amount of time. Well, in Australia, red cells are leukoreduced. That means the white blood cells are removed prior to storage. So recognition of transfused red cells depends on the recipient's immune system. Some patients are immunosuppressed and less likely to recognise transfused red cells as foreign. And not all people will make red cell antibodies and this is associated with their HLA type. So there's some patients who will never make antibodies, and we'd like to know who they are in many ways, because then that would be great. We could do that test and then we wouldn't have to cross-match the units for them. Then there are other people who make lots of antibodies, and it's interesting that in some disease states they're more prevalent than others. So patients with a hemoglobinopathy called sickle cell disease, it's hemoglobin S is the type of hemoglobin, they tend to make more antibodies. And because their cells sickle and are destroyed through that process more often, they actually require often chronic transfusion. So they're difficult to manage. The immune system recognises through all the same ways as you described in your classes as to what is foreign and what is not. It's essentially that the red cells have antigens on them. Some of them are carbohydrates, some of them are protein, some of them are waving in the breeze and be chopped off by various things and present their antigen portion there to the recipient's immune system who's out there on surveillance. Well, for routine transfusions, the ABO and RHD grouping is performed, plus an antibody screen on the patient's plasma for unexpected clinically significant antibodies against other common red blood cell antigens. And there are actually over 30 red blood cell antigen systems, and each of them has multiple numbers of alleles or antigens that are there in those categories. And I have a very nice little graph here of the blood groups who were discovered up until about 1945, when they developed the indirect antiglobulin test, which was a test in order to detect smaller numbers of immunised or sensitised the blood group systems and when they were discovered. So the first one was the ABO system, which was discovered by Landsteiner in 1901, and there were a few that were systems who were discovered, more things to have found. So that's one of the reasons there. So the protein, as I said before, they're carbohydrates. Some of them are single-pass membrane proteins, some of them are multi-pass membrane proteins, and some of them are glycoproteins as well. So there's a range of different ones, and all fascinating, and all have different antigenicity and different style of antibodies that you can detect. and different frequency in different ethnic populations, often too. There's a new markedly improved treatment for multiple myeloma, which is a blood cancer, which has just been released for use in Australia. The treatment is a therapeutic antibody which targets CD38 protein on cells. Now, CD38 is highly expressed on multiple myeloma plasma cells and has diverse functions including enzyme activity, intracellular calcium regulation, and receptor-mediated adhesion. The anti-CD38 anti-myeloma activity occurs through anti-CD38 mediated immune mechanisms, including complement-dependent cellular cytotoxicity, antibody-dependent cellular cytotoxicity, antibody-dependent cellular phagocytosis, and immunoregulatory depletion of immune-suppressive regulatory T cells. There is also direct tumoricidal activity through pro-apoptotic pathways upon cross-linking of surface CD38. It's a fabulous breakthrough, made a huge difference to the lives of patients with multiple myeloma. But the anti-CD38 also reacts with red cells and interferes with routine pre-transfusion antibody screening. And this delays provision of blood for these patients when required. So when I was working in the transfusion laboratory in the major metropolitan hospital, we collected data on the performance of this anti-CD38 in the laboratory using regular transfusion laboratory tests, and have used this information to develop strategies to manage this antibody interference to provide compatible blood and safe blood for transfusion. So some of these breakthroughs actually have an impact elsewhere that hasn't necessarily been understood by the people who actually developed the therapy. So we have been quite actively involved in this process. Well, the work of the transfusion laboratory scientists already includes support for organ transplantation, such as liver transplants, heart-lung transplants and kidney transplants. Human albumin solution, which is a blood product, is used in the manufacture of islet cells for transplantation in patients with diabetes. Blood products such as fresh frozen plasma are used in plasma exchanges to reduce the anti-A or anti-B titer levels in patients receiving ABO incompatible kidneys, and that's to reduce the chance of rejection of the transplanted kidney.